Okay, so for this lesson we're going to talk about viruses. Sections 4.1 and 2 dealt with basically how we classify things as being living or alive, and then we talked about how we classify organisms and name them. Now we're going to talk in your textbook. The third section is dealing with viruses, and they're kind of unique. So you can read about it more in your book. You can have a look at the notes and jot things down as we go through it. Add more things that will help you understand a little better. There are lots of links on the website, animations and little video lessons from other people that you might find helpful too. So let's take a look. Viruses, what are they? We know they can make us sick, but what are they actually? Some would say they're just DNA or RNA and a protein coat. And this is what some of them would typically look like. Can we classify them as living? Well, we listed a whole lot of characteristics that are required. We talked about them in sections 4.1 and 2 for something to be considered living. So among them are they must be composed of one or more cells, they must be able to reproduce, they must obtain and use energy or metabolize matter and energy, they must grow and develop, and they must respond and adapt to their environment. An organism must satisfy all of these, and there were a couple others, in order to be considered living. So, are viruses alive? The answer is no. They have no cellular structure. There's no cell membrane, cytoplasm, or organelles. They do not carry out other common life processes. There's no cell respiration, since no cell processes, and there's no need for energy, so why would they bother undergoing something like that? And they don't have the machinery to do it. In fact, they depend on a host cell in order to carry out reproduction. They can't reproduce on their own. And they cannot react to a changing environment. We do say viruses adapt, but they adapt in that they change their genetic code and then properties of their viral coat as their host builds up immunity. So they can make changes on the outside of them that can make it so, for instance, that's why you have to get a different flu shot each year, because the virus changes its coat and the old flu shots wouldn't work anymore. So, just a few little points about viruses. The word virus actually means poison in Latin. And they were first observed in 1935 with an electron microscope. They're made up of little more than DNA or RNA surrounded by a protein coat. That's why the first little statement was just DNA or RNA and a protein coat. They're often called mobile genes that parasitize cells. So parasitize. So they're like parasites. Okay, and parasites kind of live off of the host. The host does everything for it, and they don't give anything back. They don't help it, and sometimes they even kill it. So they parasitize cells. They come in a variety of shapes and sizes, but in terms of size, if you're comparing them, they are smaller than prokaryotic cells. They're 100th the size of the smallest bacterium, so that's really tiny. And bacteria and other prokaryotes are smaller than eukaryotic cells. So they're quite a bit smaller overall. They often attach to a certain receptor on hosts so they can infect the cells. And they're not active until they enter a host cell, and they may actually kind of hide in your cells or remain what we call latent in the host until an opportune time. So you may carry them around and not experience any symptoms. So you'd be a carrier, basically. They cause diseases like the measles, the mumps, AIDS. AIDS is the disease. HIV is the virus that causes AIDS. Okay? So you gotta keep those two straight. AIDS is the actual illness. HIV is the cause. It's the name of the virus. Herpes. So if you have cold sores, then cold sores are due to the herpes virus. And if you do happen to get cold sores, you'll notice that you get them sometimes and then they go away and they come back and they go away, and that's kind of the virus where it becomes active and then it kind of goes into hiding, and it becomes active and it goes into hiding. Chickenpox is also caused by a virus. The common colds, flu, hepatitis, which affects your liver. That first part of the word, hepatic, refers to the liver. So hepatitis would be infection and inflammation of the liver. And polio. And those are just a couple of pictures of some viruses. There's a few questions there that I'd like you to work on. You can read pages 122 and 123 and answer them, and we'll go over them in class. So basically, what is the function of the capsid? But I also added how is a capsid different from an envelope, I think is what I wrote. Or how does it compare? How are they related? Okay. And then how can you classify viruses? And I'd also like you to draw a bacteriophage and label those parts. So you can pause it here and you can do those questions and then you can go back. But then I'll go through them, obviously. So, classifying viruses. Viruses are classified based on their size and shape, the type of host organism they infect, on their genomes, which just means their sets of genes, their DNA or RNA, whichever they have, and their method of replication. So we're going to be looking at typical virus and how it replicates. How other ones do is similar, but not exactly the same. This one is a bacteriophage. So you're going to be drawing some of the main parts of the bacteriophage. Oh, yeah. You're supposed to sketch one. This is my sketch. Okay, very simple version of it. So, a bacteriophage is a type of virus that infects bacteria, and certain bacteriophages are specific for certain bacteria. Okay, this head, the head of the virus, consists of a protein coat or envelope called the capsid. So it's made of a whole lot of capsomeres, or little proteins that are stuck together that form the capsid, and they can look very different depending on the bacteriophage. Inside of the capsid, you would find its nucleic acid. So this one has DNA, some might have RNA. This part is the core, and these are the tail fibers. Okay, and the tail fibers are important for attachment. The core kind of holds the other parts together, but it also provides an entry passage for the nucleic acid to pass through. Okay, so that's a simple example of a virus, and it's one of the objectives that you have to be able to cover. You should be able to sketch and label, or just label a bacteriophage. So viruses can reproduce in two ways, and you should have a handout that's specific for this. They are Remember, they require a host in order to be able to reproduce. And I'm going to go through it mainly with the diagram, but these are the main points. You should be able to, we'll be doing a little demo exercise in class where you're going to work in a group, and you should be able to draw the parts and explain the main steps. Okay, tell me what's happening in each step. Okay, so when we talk about it in class, that's what we'll be doing. So you should be familiar with it so that you can help your group. So, lysis means to break apart. So in the lytic cycle of the virus, essentially the virus is going to make copies of itself and inside of the host cell, and then eventually when it's made enough copies of itself, then the cell is going to burst open. So lytic cycle is referring to the fact that it causes the cell to break open at one phase. So the first step in the lytic cycle is where the virus attaches to, so it uses its tail fibers and attaches to the host. The second step, referred to as entry, is where the virus itself, this type of virus doesn't actually enter the cell. There are some that do, but a lot will just attach to the outside. And when they talk about entry, that means where they push their chromosome, so DNA or RNA, into the host cell. So if you can imagine like a syringe, it would force its DNA from inside the capsid down through the core and then into the host cell. In the third step, the viral DNA kind of destroys the host cell's DNA and it replicates its own DNA. So it takes over the machinery of the host cell, so ribosomes, and remember the ribosomes are protein factories, and it directs those ribosomes and other enzymes to build the parts that are going to be needed to make new viruses. Okay, so in step 3, it replicates its own viral DNA, and then in step 4, it makes new proteins that are required and assembles them together to make new viruses. In the last step, in the lysis step, that's where the host cell bursts open and releases the new viruses so that they can go and repeat the process to other host cells. So they'll go and they'll infect other host cells. I'll show you the animation, I think, after we talk about the other cycle. So that is the lytic cycle. And obviously, in the lytic cycle, it's going to destroy the host cells. So if it's destroying host cells, then it's obviously going to cause some symptoms of some sort inside of the host. In this case, the bacterial cell, they're only single-celled, so it just kills them. If it was a type of virus that attacks in a similar way inside of us, then it may destroy certain cells in a certain region in your body, and that's when you'd have certain symptoms. The lysogenic cycle is the other type of cycle, and that's where basically the virus is going to incorporate itself into the host cell's DNA and kind of sit there for an opportunistic time to kind of break out and undergo the other cycle. Okay, so this first couple steps are the same. So if you look up here, I wonder if I can I don't remember that. Oh, yeah. So I have a drawing tool, that's great. So this is where it starts. We can't really see that very well. Let me try a highlighter, maybe. Nope, that doesn't work very well. No. Show up a little better. Okay, so why is that so thin? That's probably not thick. Not thick at all. What is wrong with this tool? I'm sorry. Now's not the really the time to play. There we go. We'll just make it thicker. Okay, so this is where it starts. Again, the first step is attachment. Okay, so it would attach to the host cell here, and that's not very good at all. Attach to the host cell, and then entrance happens as well. Okay, so the first step is attachment, the second step is entry, so it puts its DNA or RNA inside of the host cell. Now this is where it's either going to go the lytic path or the lysogenic path. We're going to talk about the lysogenic path this time. So in the lysogenic path, this is, oh goodness, that's really super thick now. I'm really sorry. Okay, so we're going this way. Okay, so in the lysogenic path, that viral DNA or RNA is put into the chromosome of the host. Okay, so it's inserted right into it. And we call it a provirus. And in this case, because it's a bacteriophage, they call it a prophage. So it's the same kind of thing, but it's just that they're being more specific here since the type of virus is a phage virus. So we call it a provirus or prophage because when the viral DNA or RNA is inserted into the host, then that host cell is going to start replicating like it normally would. But this time, when it replicates or makes copies of itself, it's making copies that have the viral DNA or RNA in it. Okay? So then you have a whole lot of copies of host cells that already have the viral DNA or RNA hiding in it. That is what's happening when we say that a virus is latent. It's sitting inside of the host cells, just kind of waiting for that opportune time. And so when it finds that opportune time, it will start to go the other process, the lytic cycle, which we already talked about. So in the lytic cycle, again, the host cell's DNA or RNA is going to be destroyed, and then the virus DNA or RNA is going to take over, directing the ribosomes and the other enzymes in the cell so that they can make copies of their nucleic acid and their proteins, and then they'll undergo assembly where they build new viruses, and then in the last step they would undergo the lytic part where this host cell breaks open and releases a whole lot of new viruses so they can go and infect other cells. So that's the lysogenic and the lytic cycle. So I'm just going to get rid of that and see if I can, there. So this is a good little animation to watch that explains it well. It's by McGraw Hill.
Either the lytic or the lysogenic cycle may be followed. In both cases, the first step involves the phage attaching to the host cell and injecting its DNA into the host cell. In the lytic cycle, phage nucleic acid is replicated and phage genes are expressed, resulting in production of phage proteins. Mature phage particles assemble and the host cell lyses, releasing the phage particles. In the lysogenic cycle, the phage DNA is not replicated or transcribed. Instead, the phage DNA integrates into the host cell genome. The host cell can then replicate, carrying the integrated phage genome. The integrated DNA is referred to as prophage DNA, and the host cells carrying the prophage DNA are said to be in the lysogenic state. When the cells are exposed to ultraviolet light or to certain chemicals, phage induction occurs. The prophage DNA is excised and the phage enters the lytic cycle.
So that animation was talking about, remember we said that in the lysogenic phase they don't really have an effect on the host. They're just kind of there and waiting for an opportunistic time, and sometimes that can be triggered by things in the environment, like exposure to UV or to another kind of flu if it's inside of you, or chemicals or other things. And that's just another diagram showing the steps. The last slide I was going to share about viruses is to do with the HIV virus, and that's why there's been it's not as much in the news as it has been in the past, but it's still a very real problem for some parts of the world. Most viruses are actually RNA viruses. 70%, around 70% of them are. And an example of an RNA virus is the HIV virus. And the HIV virus is a retrovirus. So that means it has to take the RNA and use a special enzyme called reverse transcriptase to undergo reverse transcription and convert RNA into DNA in order for it to be able to kind of carry on its functions inside of a host cell. So, because of this extra step in what it has to do, there's a higher chance of mutations happening, and so there's higher chance of changes on the outside of it that make it able to kind of keep on going and not be as affected by the medications and other things that we do to try and stop its infection. So because it's always changing so much, it's hard to keep ahead of it and basically get rid of it completely. So there's a couple of videos that would be really good for you to watch. They're linked right on the class website, and I'll put them in Edmodo as well. One is on how viruses infect us. That's a really good one that kind of talks about other kinds of viruses, how they infect us. So not the bacteriophages, but a typical human virus. And then the other is on HIV in particular, so you can watch a video, and they're a little bit longer. That's why I'm not showing them right in here, and I don't think I'm really supposed to show them inside of my video that I've made for you. But anyway, I will let you watch those on your own. Remember, keep picking away at your online assignments, and if you have any questions anytime, feel free to ask. Send an email, send a direct message, however you want to reach me, or even ask to meet at lunch, and I can help you understand it better.